Good afternoon, everyone. It's my great pleasure as the director of our Northwestern Physical Science Oncology Center to welcome Jan Liphart to come and speak with us today about the mechanosensitivity of tumors. You're going to see, when you see the word mechano and tumor in the same sentence, you know that you're going to get a heavy dose of physics and biology at the same time. So I'm sure that Jan is not going to disappoint. He received a bachelor's in arts from Reed College. What was your major at Reed? Atheism. Yeah. The question was, the choices are atheism or communism.
I think it was some combination of math and biochemistry.
Math and biochemistry. Excellent, excellent. And then he went on to get a PhD at Cambridge University and then was a postdoc in the Tinoco and Bustamante labs at Berkeley, kind of linking the physics and the chemistry departments at that time before he became a fellow out at Lawrence Berkeley Labs in the Physical Bioscience Division. And joined the faculty at Berkeley in the physics department in 2004. And very recently, I guess, is 2009, I guess, when you became the director of the Physical Science Oncology Center, one of 9, no, is it 12? Do we have 12? We have one of 12 in the country. So Jan leads a really fantastic team that spans several of the great campuses out on the West Coast. And I think, though, he's going to focus mostly on his specific interests in acini and tumor growth and the physical forces, how they affect gene expression and the expansion of the transformed phenotype. So, Jan, it's great to have you.
So some of what I'll be presenting, in fact, much of what I'll be presenting is still partially undigested, and so this is more of a collective effort where I hope to get your opinions and insights. Everything I'll be talking about, if it makes sense to you and you think it's interesting, and it was probably done by one of my esteemed colleagues. If you think it's a little bit shaky and premature, it was probably done by me or my lab, just as a general context. So how did I arrive at this sort of intersection of physics and cancer? What my lab does is basically three things. We like to develop new tools for precision control and measurement. And then we like to apply those tools to either complexes or machines or organelles, such as the nucleus, or we are increasingly trying to understand how cells and organized collections of cells interrogate the mechanics and architecture of their environment and how collections of cells respond to various cues from their environment. So the first thing I'll do is I'll give you just a little vignette on the single molecule biophysics, because that's really where I come from conceptually, and then we'll branch out into the big world of mechanobiology. Here's one story. Here's an example of a technology, and I'll play this movie again. What you're looking at here in green is the outline of a single nucleus, and what you're seeing as these flashing dots here are single cargos. These are quantum dots coated with synuportin alpha. And what you're looking at, if you look closely, and if this projector had a slightly higher resolution, what you would see is you would see individual cargos approach the nuclear envelope, interact with the nuclear pore complex, transition into the nucleus, and then find particular cargos in the nucleus. Let's see if we can play this. And if you look very closely, you can see that these cargos, once they've entered the nucleus, bind and unbind to certain sites. We spend a lot of time developing new microscopies, but probably if you've done that, you will have realized that the most information per unit time, per unit money, per unit sanity typically comes from probe chemistry and labeling rather than inventing fundamentally new hardware. And so this is one example where the whole secret to getting the experiment to work was to develop the coding and passivation chemistry that is required to go on the quantum dots so that they're small enough to actually be able to squeeze through the nuclear pore complex. And the benefit here of using quantum dots in this case was that we can watch a day in the life of a single cargo, all the way from the outside through the channel into the nucleus, and then find particular targets. So the kind of thing we like to do as a lab is think not so much in terms of individual componentry or any one protein. We like to come up with, we like to explain on a sort of aggregate or coarse-grained level how collections of objects might be working together to bring about certain functions. So here's that sort of thinking applied to the nuclear pore complex. So as we track these single cargos as they move through the channel back and forth, this is the outside, this is the inside, and this is time. So you're watching the cargo move through the channel, slosh back and forth, and at some point spend most of its time at the nuclear face of the NPC waiting for round GTP to come along to allow it to make this irreversible entrance into the nucleus. The whole story here can be summarized by how is it that nuclear pore complexes are able to be exquisitely selective but also very efficient. And the simple answer is it's exactly like college admissions. When you get 100,000 applications, you don't read every single one in excruciating detail. The first thing you do is apply various filters. Has this person graduated from high school? Does this person already have a college degree, and so forth? And that allows you to successively enrich for applications that you will then read in their entirety. What we find here is there's a cascade of reversible pre-filters that work together such that in aggregate, the system is rather selective. And then the irreversible step, the coupling to RAND GTP, is very much at the end. So that's where you really commit to this cargo going inside. And this is also perhaps the explanation for why the NPC doesn't clog. Right? If you had a single high affinity interaction that was the source of the specificity, that would be very easy to passivate. You know, like putting an antibody on a surface, adding the epitope, and you'd quickly end up with a biologically inert surface. But because you're dealing with a cascade of reversible filters, you perhaps can avoid clogging. So then typically once we start working on this kind of system, what we then like to do is apply these various other imaging technologies. And here's an example that's probably very hard for you to see of 3 nuclei. All the red signal is coming from every single molecule of important beta. Here is labeled with a photo-switchable tag. And what we really like to do in this context is then relate the cargos, which are in blue, to the concentrations of important beta, which is the molecule which ultimately connects, which ultimately provides directionality and specificity to transport. And this is something where, as you can tell from the images, we're very much at the stage of still figuring out how to make sense of the data and then hopefully learn more about how this coupling works. So now turning our attention to mechanobiology. So now we're going in scale from objects that have scales of maybe 100 nanometers and change, like the NPC, all the way to multicellular structures, organized multicellular structures. Part of what I'll be showing really is insights and ideas and data generated by other people, such as especially Jamie Sethian, an applied mathematician, Mina Bissell, Joe Gray, Frank McCormick, and others. So taking a big step back and thinking about cancer and cancer research and oncology, one of the high-level trends is personalization. And there are at least 2 examples, there are probably several more, of compounds that are targeted to specific molecular defects. And this is a very, very powerful idea. But as you're well aware, there's a variety of questions related to this. So why is there patient-to-patient variation? Why is it sometimes hard to make sense of genomic data? Why is it that many compounds that look very promising in high-throughput cell line screens do not work in tissue? Part of the explanation for this, one of many possible explanations, is that intracellular signaling is significantly modulated by the cell's environment. The way to illustrate this very clearly, the one picture that has made this most clear in my mind is a picture due to Timothy Newman. This was actually collected by someone else, but he drew my attention to this. And what is being done here is, this is a picture of a cell interacting with the extracellular matrix, and to make things simpler for you, I've delineated the plasma membrane with this yellow line. So Timothy did not tell me what is the cell and what is the outside. So who here can tell me what is the inside and the outside of the cell? And the point here is not so much actually figuring out what's inside and outside, but the point is to show that that's actually not so easy. And a cell is not this object floating in vacuum. Every single cell inside your body is intimately connected, chemically and mechanically, continuously, with other cells and the extracellular matrix. So whenever you study something like protein clustering, oligomerization, signal integration, binding of ligands at the plasma membrane, coupling of the cytoskeletal dynamics to internalization, endocytosis, in every single one of those cases, inside your body, every single one of those processes is intimately connected to what other cells are doing, what things like mucins are doing and various sugary molecules, and in general, what the extracellular matrix is doing. So if the inside and the outside of cells are chemically and mechanically coupled, the first question is, do mechanics matter? What I'll do now is enumerate 3 or 4 specific cases where we sort of understand what's going on, and then I'll show you a lot of stuff where we don't really understand what's going on. One classic example is T7 DNA polymerase. And you might think that T7 polymerase is really boring, right? How more boring can a polymerase be? But if you take the substrate of this polymerase and you pull on the substrate, it flips from polymerizing to being an exonuclease. So please realize that when you buy a bottle of T7 DNA polymerase, that is a statement about how you're characterizing the enzyme. That is a special zero-force statement about the enzyme. In fact, when you start playing with its substrate, it may be a polymerase or an exonuclease, depending on the substrate. Moving up in scale. Disher 2006. I take a stem cell, I put it onto something soft. What do you think it becomes? It becomes a neural progenitor cell. This has to do with, well, we'll get into that in a moment. You take the stem cell, put it onto something hard, what does it become? A bone precursor. And that is really interesting to think about, this connection between mechanics and differentiation, because it begins to answer possibly how organized structures form and things like that, how cells in your body know what tissue they're in and what they're supposed to be, and why cells sometimes may make incorrect decisions. So if you look at your body, your brain tissue, as you know, is very soft, Philadelphia cream cheese-like, if you've ever stuck your finger in to brain tissue, moving all the way to bone, which is, of course, considerably stiffer. And a lot of the experiments people do in signaling or cell biology, the cells are placed on plastic or glass, which is way, 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 way out on some extreme of this mechanics diagram here. In disease, frequently, the tissue increases its stiffness, and the normal breast is somewhere around here, and then with disease progression, it stiffens. There are many exceptions to this. For example, if you, with a large tumor, the inside, when it becomes necrotic, of course, becomes very soft and so forth. So any sort of statement like this, stiffness equals bad or something like that, is a oversimplification that has a lot of little details associated with it. So how should you begin to think about mechanics and cells and tissues and the extracellular matrix? What are you composed of? You're mostly water, right? Your number 2 constituent is extracellular matrix material. Number 3 constituent are cells. Number 4 constituent are microbes, right? What sort of picture should you have in your mind? Here's a movie from the Kumar lab, and you're seeing 3 glioma cells interacting with collagen. And if you just show this to someone who's never seen before, the first thing they think of are little spiders crawling in a spider web. And of course, as these cells are crawling through this extracellular matrix material, they're pulling on it. They have to. That's the only way to move. You have to exert a force on your substrate. And they're also sensitive to what other cells are doing around them and so forth. So that's one of the movies that is somewhat more representative of what's going on in your body right now in certain situations. So now let's think about mechanics and plasticity and phenotype and genotype. Here's a classic experiment from the Bissell lab. I take T42 cells, which are supposedly representative of a highly malignant cancer cells. I let them grow in 3D culture, and they form these arguably ugly-looking, irregular cell clusters. And now all I do is I provide a variety of soluble factors or appropriate mechanics or shear stress. And what I'm able to do, if I pick these parameters correctly, I'm able to phenotypically revert these structures. So I have the same horribly screwed up genome, mutations, amplifications, translocations, all these other things. And what I do is I provide the correct combination of external cues, and I'm able to normalize the phenotype to something that a pathologist would score as being relatively normal. The final part of this crash course in tumor mechanics and the ECM and plasticity is a 2005 experiment from Valerie's lab. What she does is take an organized structure, a mammary acinus, she places it onto increasingly stiff material, and she's able to disorganize the formerly organized structure until it looks like something that, again, a pathologist would call highly disorganized and malignant. Please forgive me, by the way, when I use medical terms incorrectly. Remember, I'm in the physics department. And so please, I'm still struggling with use of appropriate nomenclature, and so please correct me. One of the limitations of this experiment, think about it for a moment. What's really good about this experiment is that I have a defined material, I'm providing a defined signal, I put an organized structure on this, and I watch it disorganize. But is that completely reflective of what's going on in your body? And then you say, but wait, Jan, matrix metalloproteases, ECM remodeling, lysyl oxidase, right? So our extracellular matrix, for better or for worse, is something that we're constantly modifying. So one thing that's missing in this experiment conceptually, that's probably really important actually, is the ability of the structure, the multicellular structure, to modify the stuff it's sitting on, and then how those modifications in turn feed back onto behaviors of the structure itself. And that is the specific question we care about in my lab. It is possibilities of feedback of multicellular structures to the matrix and back. And this will become hopefully much more clear. So now you say, okay, if I think about signaling in a quote-unquote conventional, in terms of a standard model, I could draw particular signal pathways in this way. I have various proteins I care about. They talk to various other proteins. That leads to changes in a variety of cellular processes and decisions. So how can we begin to explain the sensitivity of things like differentiation, dedifferentiation, organization, if we think about signaling in this view? And one way to do that is to point out, which is glaringly obvious to every single person in this room, that all these molecules that participate in receiving signals, integrating signals, transmitting them into the nucleus and other things are, of course, organized in things like the plasma membrane, and many of these in turn are mechanically coupled to the extracellular matrix and mechanically coupled to the cytoskeleton, and these complexes are necessarily mechanosensitive and dynamic and all these other things. So if you think about signaling more in this way, then a lot of what you'll see now is almost trivial in some sense. It's sort of got to be that way. Okay, so now quickly concluding our introduction to mechanobiology. The first thing is we looked at examples of how the inside of the cell, this, by the way, I was kidding you, of course, this is the inside and this is the outside, is intimately connected. So cells are not these blobs of material floating in vacuum. We know that individual enzymes are exquisitely force sensitive. So if you pull on the substrate, they do things differently. And you're fortunate to have people like John Marco here and others who are interested in that sort of question and have the capabilities to answer them. Then we know that if I take an organized multicellular structure and I provide incorrect mechanical cues, I can get it to disorganize. I can get stem cells to differentiate into a variety of cell types based on mechanical cues, and we can begin to think about actual ways of how this might be working in terms of these when we take the membrane and cytoskeleton and oligomerization, spatial organization into account. The good news is that every single biologist is a secret mechanobiologist. And some of them are more open about this than others. Because realize, if you measure where something is, call it X, the position of something, if you take two pictures and you look at how that position has changed, that of course gives you dx dt. And I'm sure you're aware that F, the force, is proportional to the rate of change of position versus time, namely the acceleration. That's F equals ma. So every single biologist who takes two pictures, before and after, of how something changes position, the plasma membrane, organelles, proteins, the cytoskeleton, what they're already doing is measuring displacements, rate of change of those displacements, forces. So every biologist is a secret mechanobiologist. And my most esteemed colleagues, pathologists, who I love very much, are of course all mechanobiologists because they care about the architecture of things and how those architectures change. So please realize that if I give you a boundary and that boundary moves, it can only do that because something is pushing or pulling, or if something which is normally where two forces are balanced are no longer balanced, and then the boundary must move. So as soon as you study changes in cell shape, and changes in cell volume, changes in organelle shape, you're already measuring changes in mechanical homeostasis. You're already doing that. So pathologists are extremely good at this, which is why that's something I would like to understand much better. But I really struggle with pathology textbooks because I'm always looking for the first chapter that says, here's the theory of pathology. But instead, they normally go very quickly to many plates of examples of things. And so that's why I'm still struggling with those things. Good. And so now what I'll do in my remaining time is give you a very specific, more concrete story. We're interested here again in mammary acini. These are not individual cells. These are collections of several hundred cells. These cells are epithelial. They're polarized. And normally in the human breast, they would secrete milk into the lumen. And if you grow these exactly right, you can get them to secrete milk too, which is good. So that tells us that we're working with an object which has non-trivial biological capabilities, like making milk and secreting it into a lumen. If you want to get them to do that, you have to grow them in 3D in a basement membrane equivalent material like Matrigel. These are the structures where breast cancer typically starts. These are the structures, these are the cells most at risk for malignant transformation. And we've already mentioned the PATSEC 2005 paper. If I now take these structures and I inappropriately harden their environment, they disorganize. The first question you might have is, how come this doesn't happen all the time? One answer to that is that normally acinar structures, this is not only mammary acini, but in general, are shielded from collagen 1 by the basement membrane and my epithelial cells. So normally these mammary acini will never ever touch collagen 1. They're protected from this. And so the more specific question one can ask is, what if an acinus loses shielding from collagen 1 for whatever reason? So what we do is we grow these structures on day 8, day 9. Then we chemically strip the basement membrane. Then we let them recover in a calcium-containing buffer. We want to make sure that those cell-cell junctions are nice and tight and stable. but there's still no basement membrane, and now we take these structures and we drop them onto collagen 1. Let's just look at the movie. So here's 5 or 6 of them, and the movie's going. There's nothing in the system except collagen 1 and acini. These are RAS transformed, but it actually doesn't matter. And let me play that movie again, because it's really fast. You see them disorganized nicely. If you look closely, you see behaviors like cell spreading. You see formation of aligned collagen. And if you look very closely and quickly, you even see collagen rip right there. And so these mammary acini are able to, first of all, radially transport collagen. So this is a collection of cells radially importing collagen. And if and only if 2 of these are close enough together, then a variety of collagen architectures and mechanics can form that then have biological implications. And so that's the rest of the talk. What we like to do as a lab in general is to try to develop bespoke technologies that very directly allow us to see things which were previously hard to see. And so typically these involve mechanics or measurement of displacements or measurement of the chemical composition of an extracellular matrix as it's developing. So one of the things one would like to do in mechanics world is simply see mechanics, right? Imagine where calcium signaling would be if I couldn't see calcium. So one way one might think of doing this, and this has been done many times by many other people, is exploit birefringence. The first experiment along these lines is a 1921 experiment. A block of collagen was mechanically bent. There's a beautiful stress-induced birefringence here, and we can repeat this experiment in 2011. And again, you can all do this: buy a block of Jell-O, bend it between cross polarizers, let it relax. And if the material were rubber, which it is not at all, then it would be relatively straightforward to back out from these images what the stresses are in the material. That in general is not possible for viscoelastic materials like collagen. A lot of other things go on here. But the good news is that despite this conversion being very, very hard for viscoelastic materials like collagen, it's very easy to apply to lots of different systems. And here's a little example of a twitching zebrafish, and the signal you just saw was the strain-induced birefringence in the living tissue as the fish was twitching, right? If I take a polymeric material, it doesn't matter if it's tissue or something else. If I mechanically align it, I induce a birefringence. And so there's no labels, lasers, or anything in here. It is an inherent signal. Good, so now let's take the technology we just described and apply it to acini reorganizing collagen. So let's start, because remember, I'm in a physics department. We start with an individual hydrogen atom, and so here's our hydrogen atom, a single acinus. We let it interact with collagen. And in bright field, you see relatively little. Maybe the edges get a little bit frizzled, but that's it. If we look at this in birefringence, and this is actually structured illumination polarization microscopy, we see a birefringence form. And let's actually, I'm hopeful that given the lighting in this room and the projector, the subsequent movies will be a little bit more clear. And here's, well, I remain hopeful. Here are now 2 of them. I take 2 acini, bring them into proximity, and let's see what they do. And so let's look at this movie first. So you see 2 of them. Again, this is much better. This is a birefringence movie. And so the first thing you see that's noteworthy is if and only if 2 of them are close enough together, then they form something that a single acinus never does. It's this region of intense birefringence connecting the 2 of them. So the diffuse birefringence around here looks sort of similar to what a single acinus also does. But this region of intense birefringence in the middle is something that we think that only more than one acinus can do by working together. And this is another way of looking at the same sort of data, which is the actual transport of beads in the collagen. So this is now a bead-doped collagen, and you directly see this vector transport field as the acini are contractile. And then because of the nonlinearity of the collagen, certain patterns can form if and only if you have two of them pulling in opposite directions and they're close enough together. What do these fibers, these cables actually look like? Here's one, first acinus, second acinus, and if you look very closely, you see this region of aligned material between them, and if you look even more closely, you see regions where the collagen has been tightly bundled. So the structure actually looks something like this. And it turns out that once you begin to reorganize collagen, it becomes much less sensitive also to enzymes that degrade or remodel the extracellular matrix. So some of these enzymes are actually inhibited by particular superstructures of collagen or other fibrous material. So now let's again put on our technology hat. So we've looked at this highly simplified model system of take two acini, put them on collagen, watch it with polarization or bead transport. What if you wanted to do this in a living embryo, right? How would you actually, can we get beads in there easily? No, that's not so easy. Can we use birefringence? Yes, but ideally, to make this really simple, what you do is, for example, I wish my movies wouldn't all autoplay. But what you're looking at here is one technology where if you looked very, very quickly, you would have seen a nice simple lattice, a red lattice, that then gradually deformed, as in this case again, two acini interacted with the gel. So let's look at that again. So there's the initial lattice that is being warped. This is a very simple, again, this is an example of a new chemistry that's useful. This is a photoactivatable collagen binding protein. So what you're able to do here is you start with a blank slate, and then you write lattices in 3D into the gel, and you watch those lattices deform. So anyone with a ruler can simply back out what the strains are in the system, right? And what I'd really like to do is actually make a transgenic fish or another animal that has this, that actually secretes this material into the ECM, so I can watch development of the body plan simply through this sort of technology. The other thing you can do is, for example, write columns into the tissue and then see that column warp. But we haven't done that yet because making transgenic animals is way beyond our ken at this stage. So back to the more tractable model systems that we can actually deal with. So we've talked about a single acinus. We've talked about two. What about multiples? Again, very, very similar behavior. So they start manipulating collagen, and fibers form along geodesics, and cells stream. These cells are Ki67, high YAPTAZ in the nucleus, highly motile and other things. They then stream along these highways. And now let's zoom further out. So let's say we not only have 4 of them, but we have many more, and this sort of looks like this. This is a bright field image. Some of these guys are already disorganized. This is now a centimeter squared. And here's the underlying pattern of mechanical connectivity the acini have generated. And so again, if 2 of them are close enough and they both start pulling, then based on the nonlinearity of the material, particular structures can form. And this is one of the reasons I was actually very glad to talk to a few people here, because you are fortunate to have people who care about networks and propagation of information along networks and questions such as those, which may or may not be applicable here, but I think are certainly worth asking. The general question here is, this is pretty, but does it actually mean anything? Is it relevant at all? Remember the Patzek 2005 paper. If I put an acinus on something that's inappropriately hardened, you can begin to make a biological case for this being important. So the first question is, are these red lines actually regions of hardened collagen? We know they're aligned because otherwise there wouldn't be a birefringence signal. You can directly get at this with AFM. You can push on the cables or regions far away from the cables, as seen by the different slope. Once you align the collagen, again from a polymer physical perspective, this is not at all surprising. It's sort of got to be that way. And then you then push on that aligned material. It's effectively stiffer than if it's not aligned. Okay, fine. So it may be an AFM may possibly perceive this as being hardened, but do the cells perceive it as being hardened? So there the question is, do they translocate the mechanosensor, the YAP protein, into the nucleus? So what you see here is a confocal Z scan through a disorganizing acinus. And if you look closely, these two guys, which were sitting on a cable, nicely have the YAP ring around the nucleus. Right there, these two, and there's one, there's one. And so as soon as the cells leave the parent acinus and are moving along the collagen cables, they're also perceiving a hardened substrate. And now you can say, okay, fine, so the acini are disorganizing. The cells are sensing a hardened substrate. Still, does this matter? So here's a Kaplan-Meier analysis, and this is perhaps the first time ever a Kaplan-Meier survival plot has been applied to mammary acini. So then the question arises, okay, what's your event definition? What we've done here is we have 5 separate event definitions, and I'm not going to go through with all of them, but here's a very simple one. It's the onset of cell streaming. If I have an acinus, at which time do cells leave the acinus? The red are single acini, which are far away from all others, and the blue ones are acini that are mechanically interacting with at least one other acinus. And the average time to cell streaming extrapolated from the Kaplan-Meier curves is something like 22 hours for the blue, the interacting ones, and something like 35 hours for the red. And the chi-squared is nice. Okay, so that's an indication that there are real implications in terms of the time to streaming, the degree of disorganization. What else can we do with these sort of data? What we really want to do is relate characteristics of the extracellular matrix to much later subsequent biological, quote-unquote, biological events. So here's one way of doing this. What I'm plotting here is the bead velocity. So this is related to how fast the collagen is being remodeled very early in the experiment. And now we're waiting 14 hours, and then we're asking, or we're waiting 14 hours, 20 hours, are cells streaming or are they not? And what this is showing you is that the faster the parent acinus was remodeling the collagen early in the experiment, the faster cells begin to stream. So the p-value here is quite good. The correlation coefficient is not all that hot. The coefficient determination is like 0.15, correlation coefficient somewhere almost 0.4. So this is indicative of there's some connection between the degree to which an acinus is inappropriately engaging with the collagen 1, remodeling it, with events that then happen many, many hours later that are things like the acinus dissolves, cells stream, and so forth. Let's look more closely at these 3. This is a bead-doped experiment, and just look at this. So this guy changed its mind, right? Early on, this one was pulling really hard, and you could see the cells were almost ready to stream along this cable. But then this one said, I'm now going to pull harder, and this one now reorganized the front of disorganization, pointing to this one, and then cells began to stream along this cable. So look at that again. And one of the issues in thinking about what these interactions mean is that these cables are dynamic. So it's not like the pattern forms and it's then static. Remember, we're dealing with a viscoelastic material, And the timescales of the mechanical remodeling of the collagen are comparable to the timescales of disorganization. We think they're actually very, very closely connected. And so just because you see a cable form doesn't mean this one is always going to disorganize to this one. If this one decides to pull harder, disorganization then goes that way. So that is telling you that not only the rate, extent, dynamics of disorganization are connected to these long-range interactions. The range can be as long as a millimeter, but also any other acini in proximity, in mechanical proximity, can then change what happens in terms of the time scales and the directions and so forth. This is cell streaming. This is semi-obvious. Okay, good. So we've looked at a lot of the primary data, just the movies to see what's going on. So what you'd really like to do at this stage is ask, are we dealing with a fundamentally mechanical effect, or do we have to invoke other things that are going on here? And this is all work from the Cephian lab, our applied math collaborator. And generally what he's interested in doing is coming up with a computational framework with all the right ingredients, Navier-Stokes, advection, surface tension, all these other terms. And then he wants to change a variety of control parameters, throw these acini against a wall, stretch them, twist them, expose them to fibers or not, and open a basement membrane or create a defect and so forth. And the good news is that various algorithms are sufficiently mature these days to be able to calculate. Clearly gravity and inertia is turned up a little bit here, right? But this is just for illustration, and so it's possible to evolve in time how all these boundaries must move if mechanics are changed, if compliances are changed, and so forth. We can use those computer codes to now ask, can we recapitulate the actual structures we observe in experiment in the computer? The answer is yes, if and only if we introduce the nonlinearity of collagen. And so what collagen does, collagen is not a simple Hookean spring, right? If I pull on collagen a little bit, it's relatively soft, but then it stress hardens. So the only way to get these fibers to form is actually to introduce a stress hardening of the polymeric material. And then immediately one is able to reproduce these kind of patterns.
Is there a chemical hardening of these? Are the cells releasing diffusible enzymes that facilitate the bundling?
That's a really great question. We're struggling with that a little bit. These patterns form on timescales of 120 minutes, 3 hours, 4 hours. So that's pointing towards it being largely mechanical initially. Then, if we take a probe and we push on a region close to a fiber, we can mechanically erase the fiber as we manipulate the gel, and then if we take the probe off and let the system relax, the fiber springs back into existence. So that's telling us, at least early on, these things are not yet chemically cross-linked. However, it would not surprise me at all if I take collagen, I bundle it tightly, and I would not be surprised at all if that also has chemistry implications. And then if you looked very closely at one of the previous images, then especially this TEM image, you would have noticed rings of loss of, it's like ring around the collar, rings of loss of signal around acini. So if you look very closely, which is impossible to see, here you can see it a little bit better. You see the absence of collagen. And I hate when people do this when they give talks. They go back 40 slides, and then everyone needs to wait until they get the right slide to come up, and then their computer is too slow, and then you see their slides again for a second time. It's all super annoying, and so I apologize. Here. So this is something we don't understand. So why is there a region of depletion of collagen around the acinus? Now we're talking T equals 10 hours, 12 hours, something like that. So either the acin is pulling so hard that it's actually tearing collagen, and we know it can do that, or there's actually some enzymatic remodeling starting to kick in. But we really don't know that. And I think that's actually a really, really interesting area. So I'm the last person to say that this is all mechanics. No. What's really interesting about this is that mechanics are important, but the cells are responding, proteins are moving around. I'm sure this is changing the sort of things they're secreting. And we already know it's changing the rate at which collagen can be degraded, because as you bundle collagen, it's less sensitive to degradation and so forth. So there's all these extra connections there that I think will turn out to be really interesting. And now I'm going to do the super annoying thing again, which is to scroll forward by 15 slides. I'm going to avoid trying to do that. Okay, so the final question here is, on some level, who cares about any of this? Do you actually see evidence for this in breast cancer tissue samples? And this is an example of one of these structured illumination polarization images where the signal is a birefringence signal, so this is not proportional to the retardance. The really difficult thing we find is that any real tissue you look at has extreme heterogeneity in terms of collagen organization. So any simple pattern like highly lined collagen equals bad, it is not at all that simple, and that's one of the real challenges, right? So who's to say that this alignment right here is going to be problematic, or the wiggly pattern here and so forth. So, and there are people who are much better informed about this than I am, such as Patricia Keeley, who studies this using second harmonic imaging and so forth. So there's a lot to be learned in terms of which aspects of collagen misorganization or inappropriate microarchitecture are actually relevant clinically. So let's begin to conclude. So one thing we did is enumerate some factoids from mechanobiology, ranging from signaling to coupling of signal processing with the cytoskeleton, to single molecular machines, to organized multicellular structures, to the stem cell regenerative medicine type plasticity questions. What's the really big step? What's the really next big step? Well, what you'd really like to do is now relate all of this to all the genes and proteins and pathways that people know about in cell biology and disease. The good news is that many of these are involved in mechanosensing. ERK-B2, PGGF, P38 kinase, ERK, MEK, Notch, Wnt, Limb kinase, TGF-beta signaling, epigenetics, and other things. And it would be very interesting to start explicitly creating those connections. One person who's done that is Don Ingber, looking at VEGF signaling. And his question was, is VEGF signaling mechanosensitive? And why might that be important? And the answer is relatively clear, yes. If you modulate the mechanics of the material the cells are exposed to, the way they perceive a diffusible small molecule, the VEGF ligand, changes. And this makes sense in developmental biology, because how is a small molecule like VEGF going to ensure, how is a gradient of a small molecule going to ensure that a developing tissue is appropriately supplied with blood vessels? And part of the answer is that mechanics are modulating how the cells are perceiving the VEGF signal. This is a beautiful Nature paper, probably 2009, 2010. And now stepping way back out to explicit cancer treatment, personalization, and so forth. So everything I told you about today, you can think of in at least two ways. You can think of this as an incredible headache or a problem. Would life not be much, much easier if all these signaling pathways were not mechanosensitive, if these complexes were not connected to the cytoskeleton, if cells were not connected to the extracellular matrix, and the ECM was not modulating how cells would respond to secretable small molecules like VEGF? So that's the problem view. Or you can think about this as a great opportunity. For example, if I take a T42 cell line with all the defects it has, and I provide the right combination of external cues, some of which can be mechanical, then I can phenotypically switch that cell line into something which looks phenotypically normal. And I like to think of it as more of an opportunity rather than a big headache. Why is that? One experimental observation that's interesting is that if I establish that intracellular signaling is abnormal, the real question is, can I now normalize that abnormal intracellular signaling by not targeting the signaling itself, but by targeting the extracellular matrix? And the answer, based in part on Valerie's work, seems to be yes. For example, inhibiting collagen cross-linking normalizes high 3-AP kinase, ERK, and so forth inside the cell. And so I'm enthusiastic about this because it gives you a potentially new way of thinking about how to preventing disease and treating disease once it occurs. It's targeting the microarchitecture, microanatomy of the extracellular matrix. And so on that note, I'd like to thank all my colleagues who did everything, as we discussed earlier, that you thought was interesting was done by someone here. Sanjay, Meena, Joe, Valerie especially, and many others. Sethi and Jamie did the 3D calculations of how these boundaries change. And I'd like to thank especially the NCI for supporting a lot of this work. Thank you for your time.
Hey, are there any questions? John.
Hi, John. I was wondering, did you do experiments with the acine, this beautiful visualization of stress patterns, on some, completely non-biological type of soft gel, PDMS or something like that? Is there anything similar that we can see?
So the PatSec 2005 paper is actually on PDMS. And what you see there is, as a function of the PDMS compliance you impose, you get a different end degree of disorganization. So the harder the material, the more of a disorganized structure you observe. What I think is going on in the collagen is that there's a positive feedback loop. The acini inappropriately engages the material, mechanically concentrates and aligns it. It's stress hardened. The cell perceives this as now being on a harder material, like in PatSec 2005, disorganizes more and so forth. But then the reaction is self-limiting. Why? Because at some point the acinus flies apart. It's no longer contractile. At some point it can no longer keep manipulating the material, and cells begin to stream. The acinus literally dissolves. You end up with individual motile cells.
So I don't think you showed us a picture, maybe they didn't do the PatSec study, didn't do pairs of acini interacting. Did they look at that?
Well, in the PDMS, there is no such thing as a long-range interaction because there is no cable formation, because that requires a net transport of material, which doesn't work if you're using PDMS.
So that is specific to collagen.
It's specific, we think, to any polymeric material which can actually be transported, which is not irreversibly cross-linked. You need to enrich it, you need to concentrate the material and align it.
You know what I'm getting at is, would it work? I mean, what would be the right choice then for something that would be transportable, agarose, something transportable but not metabolizable?
Yes, and that's a very keen observation. It is, for example, possible to dope in small amounts of agarose into a collagen gel, thus preventing actual concentration of the collagen. That's an experiment we really love to do, but we're still struggling with highly reproducible initial mechanical characteristics of agarose-doped collagen. It's very tricky for us to get the material to mix them at the right time, the right concentration, and to get them to harden in exactly the right way in a reproducible manner. It's very, very easy to introduce spurious alignment of polymeric material if you change the temperatures or you do other things. You can easily end up with aligned polymeric aligned gels even if you don't want to.
But it kind of sounds like on pure agarose you would see these stress cables.
Correct. On pure agarose, they also wouldn't engage with the agarose itself because the actual engagement with the collagen is based on integrins.
Kurt.
This is beautiful work, and I really liked the image you had where the cells started streaming to one acinus and then came back and streamed to the other. And you said this one starts to pull harder. What means pull harder? Is
It's
Cracking the cells? I mean, what is that?
it is a transport velocity in micron per second. So when I say pull harder, it means that there is more processive transport of collagen underneath and in the immediate vicinity of the acinus.
But what is the cause of this? More cells in that acinus to create more force? Or, what, I guess what I'm, as a biologist, trying to understand what it is that makes the cells turn around and head back in another direction.
Well, it's the fact that one of them literally is transporting collagen more. So then your question...
So it's a property of the number of cells in that acinus then?
That I don't know. So these systems are, the only way to get some of those curves I showed and some of the statistics and numbers is do this 1,000 times, right? And so my postdocs hate me. Because the problem is that the first thing, if you do the experiment, you sprinkle the acini on the surface. Every time you end up with a different initial configuration of acini relative to other ones. Every time you do the wash, you end up with a different degree of basement membrane thinning and a different amount of defects introduced into the basement membrane. And then as they settle, different acini begin to pull at different times randomly. And we don't know what that hidden variable is. So you can have a situation where two of them start pulling early, and then a Goliath acinus starts to pull a little bit later and completely overwhelms on the other, and then it becomes the sort of nexus for all these streaming cells. But it's a great question. I don't know.
When you say pull, do you mean that it's depolymerizing collagen and then grabbing another piece and in that way? Or do you mean it's the individual cells pulling on the fiber like a railroad and like an engine moving towards the other?
That's another great question that I'd love to be answered, but I can't. So the only measurements we have is we have the collagen doped with beads, and we see beads enrich, enrich, enrich. The other measurement we have is the alignment-induced birefringence, and that increases, increases, increases. The other thing we have is the collagen labeled with the photoactivatable collagen binding protein, and that looks very similar to the B data. And then we have those TEM data. But what actually happens to the polymer, how to think about this processive radial import, I don't know. It could be, remember this is a nonlinear elastic material that can be very stiff when I pull on it, but when I try to crumple it, it's very easy to crumple. So maybe the material is just being crumpled underneath, or there's some connection between degradation and transport. And I don't know.
Rich.
What if you disrupt myosin? Does the formation of those bundles go away? In other words, is it pulling because of actin-myosin?
Yes, it's totally actomyosin contractility dependent.
So my real question was, you did talk about differential adhesion. So the integrins interact with the collagen.
Mm-hmm.
Do they interact with higher affinity to bundled collagen?
That is another really great question, and I don't know. Because the interaction of integrins with collagen and what the affinity is like and how the affinity changes the function of time and organization, it's great, but I don't know. But it's really an interesting question.
So it looks like the way the cells are growing in these spheroids is indicative of an environment that is free of serum. Is that how you were looking at this system? It didn't have any serum present?
So first, we start with a single cell, which we grow into acini in matrigel. Then at day 8 or 9, we harvest them, we spin them down, we wash the acini, thinning the basement membrane, then a calcium buffer recovery step to have the cadherins be nice and happy, and then settle onto collagen 1. So that's the overall flow of the experiment.
There is no serum present.
As far as I know, no.
You know, it's actually interesting. If you actually use serum in your experiment, that might actually change the rigidity of these polymers.
Uh-huh.
And so, having now know that you don't have serum present, it doesn't really capture what's happening in an actual environment. You have blood and serum present.
Oh, this system, please do not misinterpret anything I've said today as having any implications whatsoever for an actual tissue which has an immune system, remodeling, myoepithelial cells. This is a highly, highly simplified, somewhat thought-provoking and pretty experiment, and much remains to be done to get at that.
I understand that. I'm just saying that maybe some future experiments could be to use serum that might change the phenotype of the cells.
Ask Tom for a PSOC Travel Award, and you're welcome to come and do that very experiment.
I have a more general question. So, in general, we always think about like UV light and bad chemicals and DNA mutations that causing cancer. How much do you think? How much cancer is actually caused by like physical injury? For example, if you're beaten, because you mentioned the cells. If you get beaten, maybe the cells will be moved to a different environment. Like, for example, you mentioned these milk-producing cells in the breast.
Okay, let me rephrase your question in a way that is that is closer to the boundary of current knowledge. And one consideration might be, what is the connection between aging and normal changes in tissue compliance and composition with probability of tumors initiating in certain places but not others? So that's an interesting question. And then with respect to tumor initiation, everything you saw today are MCF10AT RAS transformed, right? And so we're not at all claiming that RAS is not important and mutations are not important. They're totally important. And the question here is, what is the interplay of genetic changes and external signals, and how do they come together or not to bring about certain behaviors?
My question is about proximity, how close or far apart do these aspects be in order to interact, driving collagen bundling, and also the other question is in the dish that you're doing this imaging on, around the edges, is that the edge effects are important?
Yeah, those are also great questions. Edge effects are really important. With respect to edge effects, there's two situations. The first one is if the collagen is too thin, then the acini, of course, sense the fact that they're only 100 microns away from a piece of sapphire or whatever it is. So that's an edge effect. And then there's the other, more literal edge effect. And the problem there is that if you've looked at the surface of a block of collagen, it's a little bit like an hourglass. And so acini tend to roll down the surface. So we always find more acini more in the middle rather than on the edges. But then if we find one that's really close to the actual plastic, it's typically highly disorganized. It's feeling that infinitely hard surface very close. And so then what is the critical distance? The critical distance is materials dependent. Realize if we take the acini and we put them into something very soft, like Matrigel, they're perfectly happy. They don't disorganize. They don't do any of this. If we do 1 mg per mil, very little. 2 mg per mil, this happens. 3 mg per mil, not much additional effect. So about 2 mg per mil, collagen 1 is where we see this happening. So these critical distances are material dependent, and they're typically on order of 1.2 millimeters. Big number if you're a 100 micron diameter multicellular structure.
So I guess some of the videos you showed kind of before you start seeing the bundling, it seemed like the acini were moving someone, or I don't know if that was I guess I wonder if the actual sort of sense of distance is longer than that, which allows them to sort of know where the other acini are and move towards them, or is it really sort of
So if you look at double acini, so these are paired, this is normalized on this axis, and the question is, how are these mechanical effects, how do they propagate through the gel? And what this is showing is the degree to which the signal decays as a function of distance. So here's acinus 2, here's acinus 1. And so what we find are decays that are consistent with a simple one-dimensional system, which is the force doesn't decay at all if I give you a 1D cable. Of course, in 2D it decays differently, in 3D it decays differently, and so forth. But some of these fibers easily propagate with little spatial attenuation over 1.2 millimeters, 1.3, 1.4 millimeters.
All right. Well, let's thank Jan for a really thought-provoking